Welcome to Neurosalience, the OHBM podcast.
On October 14th, 2022, an exciting paper was published in Science, showing convincing success in achieving, at least from my view, one of the holy grails of fMRI: clear maps and time courses of directly measured neuronal activity as it occurs at 5 milliseconds resolution. The effect follows closely the timing of spiking, and while the mechanism is not fully established, it suggested that rapid changes in T2 are caused by the neuronal membrane potential inducing reorientation of the interfacial water, as well as changes in hydration. The paper is titled In Vivo Direct Imaging of Neuronal Activity at High Temporospatial Resolution. My interview is with Professor Jang-Yeon Park, who is the senior author and advisor to graduate student and first author Phan Tan Toi. Both are at SKKU University in South Korea. Professor Park received his PhD in 2006 from the University of Minnesota. After a postdoc and position as research assistant professor at the Center for Magnetic Resonance Research at the University of Minnesota, he became assistant professor at Konkuk University in South Korea. In 2014, he started his position as associate professor at SKKU University. Phan Tan Toi, as mentioned, is Dr. Park's graduate student, having received his master's in Advanced Materials Science and Engineering from SKKU University in 2018, and bachelor's in Engineering Physics and Biomedical Engineering at Ho Chi Minh City University of Technology in 2015. This is a beautiful paper, a series of stunning experiments that provide exciting new and compelling evidence that the information in MRI still offers surprises to those who look carefully. This is a method that promises to move neuroscience and neuroimaging forward and in new directions. In this discussion, we delve into many of the experimental details, the findings, the potential caveats, the contrast mechanisms, and the possible future directions of this method for more deeply and precisely probing the brains of animal models as well as humans. So it was a great discussion. I hope you enjoy it. Okay. I first wanted to congratulate my guests, who are the first and last author on the paper. It's Phan Tan Toi and Jang-Yeon Park. Yeah, first, congratulations on a very impressive paper. This is one of the most impressive papers I've read in years, not only because of the excitement of the findings, but just simply, how beautiful the experiments were and how convincing everything was laid out. The title of this paper is In Vivo Direct Imaging of Neural Activity at High Temporal Spatial Resolution. And just to list all the authors, it's Phan Tan Toi, Hun-Jae Jang, Kyoung-Sun Min, Sung-Pil Kim, Soon-Kin Lee, Jung-Ho Lee, Ji-Young Kwag, and Jang-Yeon Park. I'm probably butchering the names, I apologize. And this just came out on October 14th, 2022. And as I mentioned, with me today are the first and last author, Phan Tan Toi and Jang-Yeon Park, who's the senior author. So just to give this paper a little bit of context, at least from my own experience, I've been trying to find direct neural activity, from my own research, at least, looking for neural current imaging, and others have been trying to do this as well. Lorentz effect imaging, also called NEMO. Other people have tried to look at neural current effects. Denis Lebihan actually looked at diffusion changes, which he claims are more rapid. That's something that others have found, others may not have found. So it's sort of up in the air a little bit. It's all very hopeful. So yeah, many of these past papers have, produced results that were intriguing, but not completely convincing, and they never quite panned out. Whereas this paper is different to me, at least. This paper was, I sort of started it being a little bit skeptical. And then as I was reading, I was like, wow, these results are convincing. You have clear maps of activation and time courses that are very clean. And it's a beautiful paper in which you also talk a little bit about mechanisms, which we'll get into. So what motivated you to undertake this project?
Toi, you want to answer the question first?
Yeah. Actually, and the first time
time when I met my professor, and like he told me about the idea of this project, and I my curiosity in neuroscience, and I curious about how the brain works, and we can kind of we matching the idea each other, and then I feel why not, why we don't try to learn about this, because at that time I was very naive and like I don't think too much about a risky. So okay, professor, I want to get this project, and let's try this. I will learn more and more. Yeah.
So as far as, I mean, also you probably had a number of projects to fall back on. I mean, just even getting this project running, you have a setup in which you can do many different things. That's impressive, at least. The idea of using a very short TE was great. And so, were you looking for neurocurrent effects, or were you looking for something else? For instance, Denis Lebihan's model with cell swelling or membrane reconfiguration. Did you feel that that was potentially something that occurred?
Yeah, okay. I think it's my turn to talk about some kind of motivation. So my motivation is actually based quite back to when I was a graduate student, for master graduate student. Not kind of abnormally is I majored in philosophy of science for
Ah,
master
okay.
degree, and at the time was very interested in complexity and also philosophical question of mind-body problem. So I was very interested in how this brain works and how this minds, comes from. And then after that, I changed it to my major to the medical physics. And as you know, I was working at a Center for Magnetic Resonance Research, University of Minnesota, supervised by Dr. Michael Goud. But I did not work on the neuroimaging stuff at the time. I worked on some
Yeah.
adiabatic pulse, something like that. But
Yes.
the CMRI is one of National Institute of Neuroscience. So there are many great researchers, in neuroscience and neuroimaging field, and many visitors and visiting scholars. So we had a lot of great seminars on this neuroimaging stuff. And whenever I listen to their amazing research result, usually based on both fMRI, I was very amazed. But I have also has a very big question in mind. Really, can we do reveal the secret of a brain using both fMRI? It's just I was not a neuroscientific researcher at the time, but I had a question in my mind, and I just thought what if we have a very high temporal resolution comparable to the duration of action potential, something like that? Because I think there are some transient effects happen there. So
Yes.
if we can follow up, come up with this kind of time scale, what would happen? But I didn't know how to do that at the time. But also I worked on the other things, analysis of theoretical analysis of adiabatic process. So I forgot that, and then after I came back to Korea, and I moved to the Songyungan University, and there are some, great scientific, neuroscience researchers around me. And then one day I had a very interesting paper, the C newspaper. There's a 2014 Nature Method paper. They used the line scan method for both the fMRI. That was very
Yep.
interesting. So, and at the time I realized, oh my God, why I didn't know, if I use this line scan scheme, I can implement this kind of milliseconds order temporal resolution as I want it.
Yes.
Oh, why I didn't know that? Oh my God, I always paid attention to the ultra fast imaging, but ultra high temporal resolution. So it was good. So I tried this millisecond order temporal resolution, and I really want to see what happens. That was first motivation. And at the time, I just thought about the underlying mechanism is as you did, and I thought about the neuronal current modeling. Okay, so even if what kind of mechanism there, if we can follow these transient effects with a similar time scale, I expected I could increase the signal sensitivity considerably. That's just
Yeah.
my gut feeling. That's it. Yeah, is it? Yeah, at the time I didn't think about the cell swelling something in detail. So observation first, and then theoretical investigation later is in this case.
And that's the best way I think a lot of things are done, where people it's just worthwhile always trying it. It's hard to predict what you'll see, and it's worth just doing it. Yeah, and I think that the idea, line scanning taken to the level of imaging, it's very similar to in cardiac imaging they have cine
Yes,
imaging,
yes,
and it's essentially that, and it's nice to know that. I mean, echoplanar imaging goes fast, and but of course the readout window is so long, and here you can obviously you shorten the readout to 5 milliseconds, and as long as you have precise timing, yeah, you can go, even though the images themselves are slow to accumulate. If you have the precise timing with the repetitions, you get extremely fast temporal resolution.
yes.
I love that story. I'm actually surprised that in the field, I think that people try things and they kind of try things certain ways and they don't see anything, and then they kind of move on to something else. And I think that this was just right. And along those lines, how long did it take you to see something?
Actually, I think as I remember, maybe 6 or 7 years ago, we just started this study, and at
Oh
the
wow.
time there's a former student, not Toi, but uh, we started this study using squid axon,
Yes.
so in vitro squid axon, but he gave up, with this topic, and he left my lab after receiving his master's degree, not PhD degree. So, and
Oh
then
no.
I met Toy, and very fortunately he accepted this, studying this project, and then he got this, he started investigating this squid axon, but at the time we got some result, but now we are not sure because it's a little bit later we found there is some RF related artifact there. So
Yeah.
now we are not sure this when the signal we got using the skid axon might be a real one or not. And after
Yeah.
that, we moved to the earthworm next year.
Oh, wow.
We already submitted that in the ISMR abstract, but it's 2019.
Yes.
So he digged, in the backyard to find the earthworm, but it was pretty tough. There was it's really hard to remove all the soil inside the earthworm. So
Oh
soil
gosh.
inside the oil, there is a metal element, so it affects the MR signal, right? There's a magnetic susceptibility effect. So it
Oh
was
god.
really tough, but he did very well. And then we moved to the red tail, mouse tail, right? And then it's an in vivo study, real mouse study, but not brain. First we studied the mouse tail, and we also
Okay.
saw something, but we did not confirm that using electrophysiology, something like that. But when we saw something, and when we use this mouse tails, experiment, oh, looks like it's working in vivo in mouse. Why don't we try to mouse brain imaging directly at the time? So I still remember 2019. There was almost end of October. This ISMR abstract deadline was about, I think it's 2 weeks before, So why don't you acquire some very preliminary data, using this occasion scheme on a mouse brain? So we
Yeah.
got very preliminary data at the time, but
Okay.
and then after that we acquired more and more data, and then just average them and group study, and then we more kind of sure, okay, there is a signal there, and then we thought about how to deal with them, and then what could what kind of thing we can do, and as like including the neural activity propagation, and what kind of experiments we need for validation, and yeah, we had a lot of discussion at the time.
Okay. So you pretty much saw something that hinted at something right from the start, it seems. And then, so why don't we just dig into the first experiments just to iterate for everybody. It was at 9.4 Tesla. It was 2D line scanning. The TR was 5 milliseconds. T was 2 milliseconds. Stimulus duration was zero point 0.5 milliseconds. And then so each line of K-space collected was 5 milliseconds. And you had, I think, a matrix size of like 72 by 54. And so how many times did you have to apply a stimulus?
The number of stimulation is exactly same as the number of phase encoding steps.
Okay, for each phase encoding step. All
Yes,
right, so that
yes.
was
Yeah.
all right. So that's and so that would be for each image it was like 72. Is or
54.
uh 54.
Yeah. Yes. 54. That's the number of phase encoding steps.
Okay, so 54, and then the whole experiment took how long? Like from start to end when you scanned? Just out of curiosity.
Yeah, actually, the experiment it lasts around few hours because we need to
Yes.
gather the animal at the beginning and we start to scan the localizer and like API for checking the mouse response, and then we go to the dynamic scan. And between the scan and scan, we also take some resting time to get the mouse take a rest a little bit. So
Yep.
totally, it take around 4 hours.
Okay, for like one scan time though, let's say you were
In this case, it's a 10.6 second.
yeah, so 10.6 seconds. That's it. That's actually what I thought I read in the paper, and I wasn't quite sure. So that's incredible. And you didn't feel that's just a okay. All right, that's great. All right, and so that those results then and used I mean, a very short TE, and obviously you were stimulating very rapidly, and so for those two reasons, it's pretty clear that there shouldn't be any bold effect. And of course, it's so slow, and it's just saturated, and it's just such a short TE. Then you were able to create these maps, and it showed that there's a clear, highly significant deviation from baseline at about 25 milliseconds, or 15 to 25 milliseconds, which was faster. It corresponded more with spiking than with the multi-unit signal. So what is it about the multi-unit signal that makes it slower and potentially not detectable by this method? Just out of
So
curiosity.
you're talking about why the LFP is slower than.
Yeah, why would the LFP be slower, and why should it be not detectable? I mean, it seems like the argument here is that it seems like temporally it corresponds to spiking.
So, to be honest, so, Peter, at this stage, we didn't figure out everything. So,
Yeah, right.
yeah, we observed that is a very interestingly, our dynamic response has a strong correlation with the spiking, spikes, not local field potential. We also was very curious about that. I think because we are still, as a not fully elucidated, the underlying mechanism, a source of signal. So someday we have a clear, more understanding of the source of signal. We might have better understanding to why this LLP is slower than Diana response. But
Yes.
as far as an LLP is kind of aggregate overall effect, right? Is a all
Yeah.
is in terms of time scale too, is including a range of time scale, something like that. So
Yeah.
this roughly is a. I'm sorry.
Yeah. And it's very convincing. I mean, temporally it does align with your spike probability, and so that was great. I mean, I actually feel that after that first experiment, you could have stopped there, and it would have been a great paper. But and that was very convincing, and of course, yeah, you mentioned your supplementary data, like various smoothing and things like that. But and you used RF spoilers to make sure that there are no residual magnetization. Just out of quick question, with your K-space lines, were you able to use navigators to align them, or was it? I mean, maybe the mouse was so well behaved and so still that you didn't need to.
Yeah, actually, in our current sequence, we didn't use the navigator process to align the case by, but maybe that's the very good idea for later like development, because maybe if we work with a human or the awake animal, there may be the motion will be much more severe. So then we can use the navigator to align the case baseline.
Yeah. Okay. All right. That's great. So,
Oh,
yeah.
Peter, can I have one additional comment on that?
Sure.
So one more thing is that you talked about the RF spooling. So when some groups want to reproduce this result, is I strongly recommend that they use a RF spooling in animal scanner too. In human scanner, it's kind of default, right? It's RF spooling. But sometimes
Yes.
write the sequence code in animal scanner. This RF spooling, we missed that very first time, but it's a this without RF spooling, sometimes it looks like there is some RF related artifact. They can be confused with the dynamic response. So I strongly recommend to use the RF spooling.
Okay. That's really good to know. And as I'm trying to think about other ways, I mean, I'm sure people will try to reproduce your results and they probably won't have exactly the configuration, but just to know those key elements like the need for RF spoiling. I'm even thinking of in terms of, like the 5 millisecond window. Is there something special about that? Like the readout window is 5 milliseconds as opposed to, for instance, if you were doing human scanning or whatever, could you do like a few more? Could you do like 10 or 15 milliseconds? And would that be okay? Because it seems like you are sampling very rapidly, almost fast enough that you could subsample a little bit.
Okay, great question. I think is the one of the reviewers also requested to see some subsample data. So actually, in one of the supplementary figures, we already provided, and we also actually want to investigate what would happen if the temporal resolution goes high. So we provided data with the PR of 5 milliseconds to the 10 millisecond, 15 millisecond. And we observed that the dynamic response goes down.
Yeah.
And I think it's the after 10 millisecond, with the longer than 10 millisecond, looks like it's going to disappear.
Interesting.
We don't know at this stage. We don't know why exactly, but so is a temporal. What we know at this stage is the temporal resolution, millisecond or the temporal resolution is a key factor to capture this kind of dynamic response.
Okay.
Yeah.
Okay. That is interesting.
That is
and that's good to know as
interesting,
well.
right?
Yeah, because you think you could. I mean, since if you just timed it just right, you could have a longer response, and maybe it's right. You're averaging over time, and that's smooth. That averages it out, but it might be more than that. There might be something else. So, that's okay. That's good to know as well. All right, so let's go on maybe to the next experiment, which was really impressive. Doing the optogenetics experiment was great. To me, that helped convince me. I mean, first, redoing the electrical stimulation of the whisker area, but then switching and then seeing the thalamus, to cortex, but then of course doing optogenetics and seeing cortex to thalamus. Yeah, I was impressed by that. The only question I had about that, and this is maybe something one worries about when they do any sort of neural imaging, is that why, if let's say you give the whisker electrical stimulation, wouldn't the signal, I mean, is there a chance the signal could propagate back and forth? I mean, you'd think that there's feedback. And so why would it necessarily, I mean, you would show this initial effect, but do you think it would reverberate? Or do you think, I mean, I'm trying to figure out whether, if there is feedback, back to the thalamus, for instance, from the cortex. And why wouldn't one see that?
Oh, actually, maybe I think you have a couple of questions there.
Yeah.
First one is this optogenetic experiment. Peter, as you know, this is very controversial topic, and many people kind of thought is maybe not possible in vivo case. I don't know. It looks like so. We thought about a lot of validations, how to validate them. So using electrophysiology or what can we do else? And then there was the optogenetic experiment, this kind of direct or further validation. So that was the motivation using this optogenetic experiment. And then
Yes.
this feedback feed forward. I'm not a I'm learning neuroscience now, so we is a yeah, thalamocortical circuit is a there's a neural activity propagation, but sometime later I don't know, might be some feedback forward between the thalamus and cortical layer. There's also feedback effect even in thalamocortical circuit, right?
Yes.
Yeah, and in case of a optogenetic experiment, when we activate these cortical neurons, and then thalamus neurons, activation, we expect that because we know that there's some feedback effect there. So when we activated cortical neurons, oh, why not? The neurons in thalamus would be activated, because there's some feedback effect in the thalamocortical circuit. So that was our first expectation, and
Yeah.
it was. So just that's it.
Yeah, that was beautiful. That was beautiful. And I imagine there's enough maybe inhibition that damps the, if there's any additional propagation, that's probably damped by maybe inhibition or whatever.
Yeah, yes. When you are right. Is that there are a lot of more experiments we can do, especially for this to investigate inhibitory action. I'm also very
Yeah.
interested in that, but it's a limited manpower. So
Yeah.
I
Yeah.
don't know
Yeah.
whether I can do that, but it's a pretty I think it's a great idea. I'm also very interested in that. Yes.
Yeah. Yeah. And okay. All right. So that, I mean, that to me actually, right. Then I saw so after I saw the optogenetic experiment, and I'm sure there's so many details that I'm missing because I'm not an expert in optogenetics, but it looked like it was just an incredible achievement to actually do that right and to actually pull out the latency, the difference in direction, which was just great. So to take it a little bit further, then, so experiment three, or the third part of the paper, I thought was sort of the validation part of the fact that it's not bold. You increase the oxygenation, and then also your mechanism looks, you suggest that it's sort of a T2, like a pure T2 effect, I guess, not susceptibility related T2, but a pure T2 effect. And I thought it was great that you had that experiment where you, with the T cells, and looking at the polarity shift, changing the membrane polarity and seeing T2 change. That was beautiful as well. A nice argument. I also am curious though. So right, you mentioned, I mean, any pure T2 effect would be accompanied by T1 effect. And I wonder, and obviously the T1 effect you mentioned in the paper takes longer, so you wouldn't see it. But do you think over time, I guess if it's only 8 seconds, you wouldn't see like a competing T1 effect, because it seems like T1 would pull down the signal, whereas T2 would increase it. Um, so yeah, but there is, what's interesting to me is that if you scan longer, I mean, I don't know, I'm just already thinking ahead in terms of human experiments. There is a T1 effect there. It's just that it's not transient, but it might accumulate over time. If you didn't have the speed to see this effect, you might be able to see a T1 effect as well.
Yeah, absolutely. You you have a great point. You're right. First of all, about regarding the source of this dynamic mechanism, we had a lot of discussion, especially with my colleague, Dr. Seung-Kyun Lee. So we had a lot of discussion because we observed something, but based on neuronal current modeling, we couldn't explain that. Because so we simulated Lorentz force and theoretical investigation, simulation. We investigate every possibility, but was not enough to explain this signal percent signal change in vivo situation. So
Yes.
then we one day we reviewed some paper, 2018 Nature Communication paper did there. They used the second harmonic wave imaging to investigate the membrane potential change of a single neuronal cell. So
Yes.
they talked about some bound water, some molecules, change of bound water molecules can be reflected in, to detect the changes in membrane potential. That was the starting point. Okay, then if we can measure that in MRI, is MRI measurable? Maybe that might be a source of Diana signal. That was the starting point. And then, as you said, there was a T1 T2 effect there. and they are kind of conflict each other. So that was kind of was bothering us at the time. But when we simulate that, even if they are conflicting each other, still according to our simulation, we could explain this kind of a diana percent change, according based on simulation. We also provided that in supplementary material. And also one more argument is from the reviewer is the T1 effect. T1 is always has the order of 100 milliseconds.
Yeah.
Yeah, right, on the order of it. So it takes some time to manifest itself in MR signal, even if there is a T1 change there. So in this kind of case, it's a very high temporal resolution. It may not be easy to manifest itself. So we did the experiment, T2, T1 weighting, T2 weighting experiment, So we changed the flip angle, we changed the TE, and we kind of saw that we could hardly see the T1 dependence in diurnal response, and we could see the T2 dependence as you see in the supplementary material. And as you told me, in human case, we might have a more longer intersimulation period. We might need to consider this T1 effect more seriously. I think other smart researchers will help us, I think.
No, it's really intriguing. And do you think that, so if it happens at the membrane, do you think there's actually, and then I thought, well, relative to the total tissue content in a voxel, for instance, do you think there's enough tissue?
At the very beginning, we did a lot of simulation, but the Kyung-Sun Min, the student, another student, we worked on this, underlying mechanism together. And at the time, he did a simulation based on the, neuronal density and, the amount of the portion of the bound water molecules. And then we modeled the two compartment models, free water, bound water. And then when we simulate that, there we got a similar kind of result to our experimental result. So, it's very, very preliminary simulation, but gave us a kind of some, strong motivation, to drive this bound water thing, But still, when we did the T-cell experiment, we are still doing that, and we are preparing the paper. So we also see some kind of cell swelling effect, non-negligible. So we mentioned the cell swelling as well as the some changes in bound water molecules, both of them. So but the T-cell experiment has a limitation because it's not a transient effect. We changed the membrane potential once and for all. So there was some logical gap between them, but still, I think there's some contribution from both of them. Now we want to evaluate the contribution of each of them now.
Yeah, you're right. I mean, it's not a final argument, but it's a lot of compelling evidence that this might be the mechanism. Do you think that? And it's funny when on Twitter I was posted this, and a lot of people gave feedback, and Denis Lebihan was one of the people who gave feedback saying, "Oh, this is what I'm looking. This is what I see with my diffusion weighting." So do you see? Is there a link?
So I'm really impressed by the Dr. Lebihan's PNAS paper 2006. Know that the fast and detection of neuronal activation with the diffusion MRI, right?
Yep.
There he provided the explanation for the rapid response of all the neuronal activation, and he talked about this membrane bound water molecules, the contribution of that. So I was surprised when I saw that because we approached this one, like I told you, some changes in bound water molecules may contribute to this diurnal, signal and something like that. And also one of the reviewers, diffusion MRI they also address the direct detection of neuronal activity, they insist, and there are great achievements there. So when I read his paper, oh my God, okay, right? We have some kind of link this point in terms of
Yeah.
some signal source, and
Yes.
to detect the neural activity using MRI. So one side from the diffusion MRI is another side from us. So maybe
Right.
we are approaching the same thing
Yeah.
from the different side. But what I'm just wondering is this diffusion MRI has some limitation in temporal resolution, right? Is they apply
Yes.
to diffusion gradient. So maybe I think if they can reduce this TR less than 10 milliseconds, something like that. I think is that they might find something much more interesting. But as an MR physicist, that might not be very easy, but is that is my expectation.
Right. I mean, obviously, that would take some awfully strong gradients to do that so quickly. But it is interesting to suggest that maybe it's some similar effect, but manifest on a slower time scale in some way with diffusion, and manifests easily seeable at this time scale in this way. And it's also interesting, I think, that since you're going so fast, right, you don't really have to worry about bold quite as much, but it seems like you could use a slightly longer TE to be a little bit more sensitive to T2
Sure,
potentially. I mean,
sure.
um, yeah,
That's the another, yeah. There's a trade-off. We want to increase the signal sensitivity. We know that this is T2 weighting, so maybe you can increase the TE, and then we may have a more higher signal sensitivity, right? But when we increase the TE, we have to increase the TR too, right? So
yeah,
then
yeah,
we lose the temporal resolution. So there's a trade-off there. But
yes,
maybe some way to increase this, T2 weighting, not
yeah,
increasing, is not
right.
increasing the TR, is something like that.
Yeah, but then you would have to have the prep. The prep, it would have to be fast because it's affecting whatever is changing that transiently. So it might not be that might not work either. Yeah, it's interesting to think about.
We also talk about the prep. If somebody say, you know, this study is a proof of concept. So I think that there are a lot of things, we need to do, after this paper.
Yeah, one other question about contrast mechanisms, though. Just I just want to quickly finish, and then we can talk about the future. And that is another thing on Twitter. Another question was, oh, is there any chance of ruling out inflow effects? I mean, obviously the inflow effects would be really slow, but there might be some fast inflow that we don't know about. Is that a concern?
Yeah, of course. Always this inflow effect. It's been always bothering us, but we were able to see some clear effect, and then where is the inflow effect? And then one thing was we applied a single slice with a very thin single slice. That was kind of one relief. Another thing is we suggested how to suppress the body effect, right? Hemodynamic response. There are two aspects. One is from the acquisition scheme. This kind of fast, the interstimulation period is quite shorter than the hemodynamic response on the
Yeah.
order of second scale. So
Yes.
we thought this inflow effect is also that's related to this perfusion and this hemodynamic
Yes.
like a hemodynamic response relatively much longer than this acquisition scheme. So when we simulate that kind of situation, even if there is some change like a inflow effect or hemodynamic response, something like that, we could capture this kind of fast event synchronized effect. That was
Yes.
our answer at this stage.
That's interesting. That's yeah.
Yeah,
So
but
so
we
right.
need to figure it out more. I don't know.
I mean, if it is, I mean, right. I mean, in the bottom line is that you have a robust effect and it's rapid and it seems like it's temporally correlated with spiking. I think that it seems straightforward to rule out different types of inflow effects or
Yeah, of course. It's, you're right. You're right, Peter.
that was really convincing. And so the last experiment that I saw, I mean, I'm not going to go into too much detail, but I was then, I thought, okay, now I'm done. And then I kept on reading and like, oh my gosh, they're doing layer effects and looking at the propagation of activity from the middle layers through the upper and lower layers, and corresponding that with depth electrodes, and that was. Then I was just like, oh my gosh, this is just like. So this, this is probably your whole PhD thesis right here, right? I mean, you can be like, you can have
going to have a final defense. With only this one paper.
Yeah, exactly. This one paper. I think I remember a story about this one guy, this one physicist, de Broglie, who had like a 2-page paper or something, or 2-page thesis, and this would be close. But yeah, so that experiment. I mean, I was certainly impressed with the tracking of the temporal onset across layers. That was super impressive. But then what really was really exciting to me, that shows something that electrophysiology can't see as easily, and that was the suggestion that you were seeing hyperpolarization early on with an inhibitory wave, and that would open up all kinds of things.
Yes.
In neuroscience. What did you think of that when you saw that? I mean, how that must have been exciting.
There is something funny behind the story here. Actually, we analyzed this figure 5, and we kind of have some preliminary analytical data, but we didn't include this one at this first manuscript. As you already saw in the bioRxiv manuscript,
Yes.
we didn't include that, but the editor requested what you really could do with this amazing technique. What you could do something challenging, show me that. And so, okay, we have to include this one because it's. And then we analyzed that, and also at the time we also found that because from the T-cell experiment, we knew that there's a hyperpolarization and depolarization. There is a linear T2 correlation between membrane potential change and T2. So why not? Why do we could see this effect
Yes.
as a negative signal change, and then we saw that, and I discussed with Professor Park, another corresponding author. She's a neuroscientist, great neuroscientist, and then she was very amazed, and wow. Could we see this inhibitory action? Oh my God, could be a really really big thing.
Yes.
She's using electroneurophysiology.
Yeah.
Even she couldn't do that, and
Right.
so
Yeah.
so she was very amazed, and so wow, we are very happy to hear that. Okay, so everything is consistent with the previous study. Our result, looks like. Oh my God! We did something. Oh my God! We have something here. So
that must have been exciting. That whole process of having this, and then you put it in bioRxiv, definitely, but the impact in science was I think now. I mean, I have people emailing me because they know I think about this sort of thing, and they're asking me, "What do you think of this paper? What do you think of this?" So right.
I really appreciate the editor.
Yeah, that was a good idea by the editor, without a doubt.
Yeah, it was Dr. Peter Stern. Oh, he's great. Yeah.
Yeah, that was really good. So then, okay, so now that the paper's out and everyone's excited, they're all ready. Even in my group, I have a postdoc who's working on, we have a sequence like this working. We're seeing what we can do in human studies. And like you mentioned, the critical thing is probably that 5 millisecond window. I really liked your discussion in your supplementary material about applying this to humans. And one thing I didn't realize is that you mentioned that rat neuronal density is higher than in humans, and so you have a boost in potentially the signal there. But of course, obviously, if you use larger voxels, up to 7 Tesla, if you have navigators for the K space. It would work. The only thing that I think is missing, and the only way we can get at this would be if you did like medial nerve stimulation. Potentially, that would be more precise timing than visual. I mean, maybe visual stimulation would be great, but it would be really hard, I think, unless there was some other way that didn't rely on seeing the average transient. If you could look at somehow the noise in the signal, as a measure, as a metric for neural activity. Like a mass effect of, when people look at spiking, sometimes they look at just the average effect, and if you have more noise in the signal, that might show up as, that might be indicative of neural activity in some regard.
First of all, in terms of the neuronal density, you're right. So actually, I gave a rough theoretical estimation about the human translation of Diana in a supplementary text, second text.
Yes.
But
yeah, that was good. I like that. Yeah.
Actually, you are right. Is that the density of the mouse is higher than human, but in case of human, the voxel size is much bigger than the in mouse case. But that is good or bad. We cannot say that clearly here because we have a strong correlation with spikes. But spikes as a kind of stochastically, occur. So
Right.
the voxel size is getting larger. It's a signal is bigger, but it's always good. We are not sure at this point. I think it's with better, more higher resolution, smaller voxel size might be better. We are looking for some optimal size. I've been also working on this human translation now. And so, according to my theoretical speculation, we could do that in human study. So that's why I studied the human studies from a year ago. And I'm pretty sure we could see something very clearly at 7 Tesla. But, I also want to use it, but it was not accessible right now. So I'm trying to use that very soon. But so that's it. So there are many things to talk about this one. Very recently, I found some very interesting paper thanks to my another student, Kyung-Sun Min. There is some invariance in current dipole moment in different species. So what I mean is the signal source of Diana might not be proportional to the neuronal density itself. What I mean is or rather, it might be proportional to the surface charge density, not the volume. Yeah. So in that case, the signal sensitivity in human scanner, what I expected from my theoretical speculation, may be much more reduced. But still, when I calculate that, still could see even at 3T. But if 3T is maybe like at 9.4 Tesla, the kind of 0.23% from very small, but I still believe it could see that. But 7 Tesla, maybe more likely to see the Diana effect. That's what I expect. Yes.
Yeah. Obviously, there'll be a lot of people trying, and there'll be a lot of people trying different types of experiments. We're definitely considering this as well, and certainly, motion effects and things like that would be more of a fact.
Sure, I'm very sorry, but I have to add one more comment on the previous thing. The paper I mentioned about the invariance in current dipole moment thing. The paper originally is suggested by the Dr. Padma Sundaram and at Harvard Medical School. So she sent me an email about the underlying mechanism. So yeah, I forgot to say her name. So maybe she heard this podcast. Oh my God, I sent that paper to him, and so.
Well, that's good that you acknowledged.
Thanks, Padma. Okay, so.
I mean, all of these things are sort of like there's ideas all over the place. There's good ideas, and it's a matter of bringing them together in the right model. And do you think that, like in 5 years or 10 years, this will be used a lot by maybe neuroscientists that just work in animal models, or do you think that it might be used by cognitive neuroscientists with humans, but maybe not necessarily replacing bold, but maybe complementing bold in some regard?
Yeah, I know there is a very wonderful and beautiful legacy of bold fMRI. I think we inherit this legacy of bold fMRI, and so might be I don't know, it's a replaceable but I think it's better be complementary. There might be a more beautiful picture I
Yeah.
have imagined. It's the amazing work so far. In using bold fMRI. I know that, I respect them, but I know it's many people will have a better kind of a future research direction using Diana, but I have a big direction in two directions in my mind, and using animal MRI is a I'm preparing some disease model, mouse disease model, so with a neurodegenerative disease such as Alzheimer's disease and Parkinson disease. So I want to investigate some cognitive impairment in this kind of neurodegenerative disease. And also I want to investigate some psychiatric disorder too. So maybe, not maybe, I hope is Diana can contribute to the more quantitative and accurate evaluation of this kind of psychiatric disorder or some cognitive impairment, something like that, from a
Yeah.
clinical standpoint. So that's
Huh.
that's one study, and if it's working well, I also think about translation to human study too. But another one is the kind of with together other researchers all over the world, and I really want to do a real functional connection project, from mouse to human. Is a actually using Diana. I think I expect is we can investigate the causal or hierarchical functional connectivity of the in brain networks.
Yes.
So yeah,
Yes.
that's I've been always curious about, and so how this real brain network is really working, how they propagate this information, and how to deal with it. And as I told you, and as I majored in philosophy of science, and so someday I really want to know is how this mind really arises from so from this kind of network. And so I've been always thinking about that, even if I've
Yeah.
been working on other things. So what kind of properties of this network can kind of make this kind of mind? So connect them and then figure out the dynamic network, and then real maybe it can give a good insight to the artificial neural network people too, I think.
Yeah. So what you said generated two questions in my mind. So one, so you're able to see cortex, like the thalamus and cortex, but when you talk about propagation, as far as the white matter track, were you able to see, do you think that you could be able to see anything like, for instance, along the white matter track? Or
That was a great question, and we also expected to see that, along the white matter tract.
yeah.
So that's the why we did the mouse tail experiment. But
Yes.
according to our experience, looks like we could see some synaptic regions, along the white matter track. We couldn't see that. So axon potential themselves, along the white matter track, axon, there might be
Yes.
not good enough signal sensitivity at this stage. I don't know, much more higher, ultra high, magnetic field or something like that. I don't know, but it's for now we couldn't see that along the
Yeah.
white matter tract.
And right, it might be, the propagation might be quite narrow relative to the cortex, so it might not be
Yes,
much
yes,
tissue there that's
yes.
doing that. That's interesting. And the other question, so you mentioned applying it to cognitive disorders. I'm just trying to get a sense of what that experiment might look like.
I see they evaluate their cognitive impairment like some kind of exam, or ask question and answers. But what I expect is when we're looking at so apply some kind of stimulation, whatever is a visual or some kind of stimulation, we need to look for that. But and then how this networks, like maybe, for example, the somatosensory network, there's some kind of response delay there, or some amplitude changes, or take some root, or there's some different in feed forward response.
Yeah.
Yeah,
Yeah.
including all of these things. That's so I also want to make some good model to explain this kind of dynamic neuronal network.
Yeah.
That may be very helpful.
Yeah, and one random idea as well. It just also occurred to me as I was looking at your paper that there's a lot of people are trying to look at mechanisms for how transcranial stimulation works. I mean, so you could actually, if you apply transcranial direct current stimulation, one possible mechanism is that it changes the set point of the excitability, or changes the polarity, just a little bit. And it seems like this would be something that could detect a shift in it using Diana potentially with this.
That's a beautiful idea. I think it's for the validation of Diana, this in terms of hyperpolarization and depolarization, inhibitory actions, right? I think it's a beautiful idea. Yeah, right.
So this is very exciting. I'm kind of curious what your plans are, Toi.
Yes, sure. Thank you for the question. Actually, I'm still continuing doing this project with another stimulation and kind of upgrade version of the Diana sequence, and I also involve a little bit into the human study so far because it's exciting me. I cannot give up. I need to continue this.
Yeah. Well, it's actually nice because you have a result and you have something to go on, and then and it's just a matter of sort of delving more into it to figure out what it is, how you can refine the sequences, maybe to do it better. There's all kinds of really cool directions to take this. And that's the thing. It seems like a small signal, but it's robust if you do it right. It seems like it works, and so. I'm very impressed, and I don't know if there's anything you wanted to add beyond this, but overall, I think that this was helpful in terms of getting a sense of some of the aspects of the experiment. I don't know if there's anything.
I think we discussed most of the things we need to do enough, and then I just recommend people to read supplementary materials too. There are a lot of figures, we provided as much as possible, so they might be helpful for the research, I think. So, and also, Peter, my honor and very pleased to speak with you today. And you interview me, and it's a kind of special meaning to me. I know you've been very interested in this topic for a long time, so I respect it, and I really like your review paper, the title, detection of a neuronal activity, fantasy, possibility, reality. I was fascinated by the title, and I really liked that, and so it was really great. I'm very thankful to you. Thank you very much.
Well, thank you, and definitely. I think that it's wonderful that there's so much expertise that went into this, but most importantly, I think the idea of just trying these things. I mean, I know so many people who sort of they sort of talk themselves out of experiments. They're like, oh, that's not going to work because it's not this and just trying these things, I think, is the key to advancing. And certainly if you've excited the entire field of at least neuroimaging and many neuroscientists from this paper. And so we're all excited. We all want it to work, and we'll all do our best to see if we can make it propagate and work better. And it's a wonderful tool.
Again, is just inheriting the beautiful and wonderful legacy of bold fMRI. I just want to make a big leap forward, with other colleagues and other, community people together. And that's what I really hope. And
Okay. Yeah. Well, thank you both. This was a great discussion. Hopefully it will be helpful to all the people listening and watching. It's actually going to be on YouTube as well.
Yeah. So please, this is our first time to join the podcast.
Well, it's a growing audience. No, this was perfect. This was really helpful.
Okay, great.
Yeah, it was really good. And once again, congratulations to both you, and look forward to seeing you at some meeting presenting more data.
Of course. So, thank you very much again for inviting us. Thank you very much for
everything. Thank you very much.
Take for your time.
Okay, thank you.
Yeah.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Ömer Faruk Gülban and Jeff Mensch.